Reptiles are tetrapod animals in the class Reptilia, comprising today's turtles, crocodilians, snakes, amphisbaenians, lizards, tuatara, and their extinct relatives. The study of these traditional reptile orders, historically combined with that of modern amphibians, is called herpetology, because some reptiles are more closely related to birds than they are to other reptiles. E.g., crocodiles are more closely related to birds than they are to lizards. The traditional groups of reptiles listed above do not together constitute a monophyletic grouping, or clade. For this reason, many modern scientists prefer to consider the birds part of Reptilia as well, thereby making Reptilia a monophyletic class. The earliest known proto-reptiles originated around 312 million years ago during the Carboniferous period, having evolved from advanced reptiliomorph tetrapods that became increasingly adapted to life on dry land. Some early examples include the lizard-like Hylonomus and Casineria. In addition to the living reptiles, there are many diverse groups that are now extinct, in some cases due to mass extinction events. In particular, the Cretaceous Paleogene extinction event wiped out the pterosaurs, plesiosaurs, ornithischians, and sauropods, as well as many species of theropods, e.g., Tyrannosaurus, Velociraptor, Spinosaurus, Allosaurus, and birds, crocodiliforms, and squamates, e.g., mosasaurids. Modern non-avian reptiles inhabit every continent with the exception of Antarctica. If birds are classed as reptiles, then all continents are inhabited. Several living subgroups are recognized: testudines, turtles and tortoises, approximately 400 species; Rhynchocephalia, tuatara from New Zealand, 1 species; squamata, lizards, snakes, and worm lizards, over 9,600 species; crocodilia, crocodiles, gavials, caimans, and alligators, 25 species; and aves, birds, 10,000 species. Reptiles are tetrapod vertebrates, creatures that either have 4 limbs or, like snakes, are descended from 4-limbed ancestors. Unlike amphibians, reptiles do not have an aquatic larval stage. Most reptiles are oviparous, although several species of squamates are viviparous, as were some extinct aquatic clades. The fetus develops within the mother, contained in a placenta rather than an eggshell. As amniotes, reptile eggs are surrounded by membranes for protection and transport, which adapts them to reproduction on dry land. Many of the viviparous species feed their fetuses through various forms of placenta analogous to those of mammals, with some providing initial care for their hatchlings. Extant reptiles range in size from a tiny gecko, Sphaerodactylus ariasi, which can grow up to 17 mm (0.7 in), to the saltwater crocodile, Crocodilus porosus, which may reach 6 meters (19.7 feet) in length and weigh over 1,000 kilograms (2,200 pounds). Classification. Research history. In the 18th century, the reptiles were, from the outset of classification, grouped with the amphibians. Linnaeus, working from species poor Sweden, where the common adder and grass snake are often found hunting in water, included all reptiles and amphibians in class 3, Amphibia, in his Systema Naturae. The terms a reptile and amphibian were largely interchangeable. Reptile, from Latin repere, to creep, being preferred by the French. Josephus Nicolaus Laurenti was the first to formally use the term reptilia for an expanded selection of reptiles and amphibians basically similar to that of Linnaeus. Today, the two groups are still commonly treated under the same heading as herptiles. It was not until the beginning of the 19th century that it became clear that reptiles and amphibians are, in fact, quite different animals, and Pierre-André Littré erected the class Batrachia, 1825, for the latter, dividing the tetrapods into the four familiar classes of reptiles, amphibians, birds, and mammals. The British anatomist Thomas Henry Huxley made Littré's definition popular and, together with Richard Owen, expanded Reptilia to include the various fossil, antediluvian monsters, including dinosaurs and the mammal-like synapsid Dicynodon he helped describe. This was not the only possible classification scheme. In the Hunterian lectures delivered at the Royal College of Surgeons in 1863, Huxley grouped the vertebrates into mammals, sauroids, and ichthyoids, the latter containing the fishes and amphibians. He subsequently proposed the names of Sauropsida and Ichthyopsida for the latter two groups. In 1866, Haeckel demonstrated that vertebrates could be divided based on their reproductive strategies, and that reptiles, birds, and mammals were united by the amniotic egg. The terms Sauropsida, lizard faces, and theropsida, beast faces, were used again in 1916 by E. S. Goodrich to distinguish between lizards, birds, and their relatives on the one hand, Sauropsida, and mammals and their extinct relatives, theropsida, on the other. Goodrich supported this division by the nature of the hearts and blood vessels in each group, and other features, such as the structure of the forebrain. According to Goodrich, both lineages evolved from an earlier stem group, Protosauria, first lizards, in which he included some animals today considered reptile-like amphibians, as well as early reptiles. In 1956, D.M. S. Watson observed that the first two groups diverged very early in reptilian history, so he divided Goodrich's Protosauria between them. He also reinterpreted Sauropsida and Theropsida to exclude birds and mammals, respectively. Thus his Sauropsida included Procolophoniae, Eosuchia, Melarosauria, Chelonia, turtles, Squamata, lizards and snakes, Rhynchocephalia, Crocodilia, Thecodonts, paraphyletic basal Archosauria, non-avian dinosaurs, pterosaurs, ichthyosaurs, and sauropterygians. In the late 19th century, a number of definitions of reptilia were offered. The traits listed by Lydecker in 1896, for example, include a single occipital condyle, a jaw joint formed by the quadrate and articular bones, and certain characteristics of the vertebrae. The animals singled out by these formulations, the amniotes other than the mammals and the birds, are still those considered reptiles today. The synapsad sauropsid division supplemented another approach, one that split the reptiles into four subclasses based on the number and position of temporal fenestrae, openings in the sides of the skull behind the eyes. This classification was initiated by Henri Fairfield Osborn and elaborated and made popular by Romer's classic vertebrate paleontology. Those four subclasses were Anapsida, no fenestra, cotylosaurs and chelonia, turtles and relatives; Synapsida, one low fenestra, pelycosaurs and theropsids, the mammal-like reptiles; Euryapsida, one high fenestra, above the postorbital and squamosal, protosaurs, small, early lizard-like reptiles; and the marine sauropterygians and ichthyosaurs, the latter called Parasida in Osborn's work. Dipsida, two fenestra, most reptiles, including lizards, snakes, crocodilians, dinosaurs and pterosaurs. The composition of Euryapsida was uncertain. Ichthyosaurs were, at times, considered to have arisen independently of the other Euryapsids. and given the older name Parasida. Parasida was later discarded as a group for the most part. Ichthyosaurs being classified as Incerticedae with Euryapsida. However, 4 or 3 if Euryapsida is sunk into Dipsida. Subclasses remained more or less universal for non-specialist work throughout the 20th century. It has largely been abandoned by recent researchers. In particular, the anapsid condition has been found to occur so variably among unrelated groups that it is not now considered a useful distinction. Phylogenetics and modern definition. By the early 21st century, vertebrate paleontologists were beginning to adopt phylogenetic taxonomy, in which all groups are defined in such a way as to be monophyletic. That is, groups include all descendants of a particular ancestor. The reptiles as historically defined are paraphyletic, since they exclude both birds and mammals. These respectively evolved from dinosaurs and from early theropods, which were both traditionally called reptiles. Birds are more closely related to crocodilians than the latter are to the rest of extant reptiles. Colin Tudge wrote, "Mammals are a clade, and therefore the cladders are happy to acknowledge the traditional taxon Mammalia. And birds, too, are a clade, universally ascribed to the formal taxon Aves. Mammalia and Aves are, in fact, subclades within the grand clade of the Amniota. But the traditional class Reptilia is not a clade. It is just a section of the clade Amniota. The section that is left after the Mammalia and Aves have been hived off. It cannot be defined by synapomorphies, as is the proper way. Instead, it is defined by a combination of the features it has and the features it lacks. Reptiles are the amniots that lack fur or feathers. At best, the cladists suggest, we could say that the traditional Reptilia are non-avian, non-mammalian amniots, despite the early proposals for replacing the paraphyletic Reptilia with a monophyletic Sauropsida, which includes birds. That term was never adopted widely or, when it was not applied consistently. When Sauropsida was used, it often had the same content or even the same definition as Reptilia. In 1988, Jacques Gauthier proposed a cladistic definition of Reptilia as a monophyletic node-based crown group containing turtles, lizards and snakes, crocodilians, and birds, their common ancestor and all its descendants. Because the actual relationship of turtles to other reptiles was not yet well understood at this time, Gauthier's definition came to be considered inadequate. A variety of other definitions were proposed by other scientists in the years following Gauthier's paper. The first such new definition, which attempted to adhere to the standards of the Phylocode, was published by Modesto and Anderson, Indiana 2004. Modesto and Anderson reviewed the many previous definitions and proposed a modified definition, which they intended to retain most traditional content of the group while keeping it stable and monophyletic. They defined Reptilia as all amniots closer to Lacerta agilis and Crocodilus niloticus than to Homo sapiens. This stem-based definition is equivalent to the more common definition of Sauropsida, which Modesto and Anderson synonymized with Reptilia, since the latter is better known and more frequently used. Unlike most previous definitions of Reptilia, however, Modesto and Anderson's definition includes birds, as they are within the clade that includes both lizards and crocodiles. Taxonomy. Classification 2 order level of the reptiles. After Benton, 2014. Class Reptilia. Subclass Parareptilia. Order Parasauromorpha. Subclass Eureptilia. Infraclass Dipsida. Order Younginiforms. Infraclass Neodiapsida. Order Testudinata. Turtles. Infraclass Lepidosauromorpha. Infrasubclass Unnamed. Infraclass Ichthyosauria, order Thalattosauria, superorder Lepidosauriformes, order Rhynchocephalia, tuatara, order Squamata, lizards and snakes. Infrasubclass Sauropterygia, order Plecodontia, order Eosauripterygia, order Plesiosauria. Infraclass Archosauromorpha, order Rhynchosauria, order Proterosaurs, order Phytosauria. Division Archosauriformes, subdivision Archosauria, superorder Crocodiliomorpha, order Crocodilia. Infradivision Avemetatarsalia, infrasubdivision Ornithischians, order Pterosauria, superorder Dinosauria, order Sauropods, INCL. Clade Aves, order Ornithischians, phylogeny. The cladogram presented here illustrates the family tree of reptiles and follows a simplified version of the relationships found by M. S. Lee in 2013. All genetic studies have supported the hypothesis that turtles are diapsids. Some have placed turtles within Archosauriformes, though a few have recovered turtles as Lepidosauriformes instead. The cladogram below used a combination of genetic, molecular, and fossil, morphological data to obtain its results. The position of turtles. The placement of turtles has historically been highly variable. Classically, turtles were considered to be related to the primitive anapsid reptiles. Molecular work has usually placed turtles within the diapsids. So far 3 turtle genomes have been sequenced. The results placed turtles as a sister clade to the archosaurs, the group that includes crocodiles, dinosaurs, and birds. Evolutionary history. Origin of the reptiles. The origin of the reptiles lies about 310 to 320 million years ago, in the steaming swamps of the late Carboniferous period, when the first reptiles evolved from advanced reptiliomorphs. The oldest known animal that may have been an amniot is Casineria, though it may have been a temnospondyle. A series of footprints from the fossil strata of Nova Scotia dated to 315 marsh show typical reptilian toes and imprints of scales. These tracks are attributed to Hylonomus, the oldest unquestionable reptile known. It was a small, lizard-like animal, about 20 to 30 centimeters, 7.9 to 11.8 in, long, with numerous sharp teeth indicating an insectivorous diet. Other examples include West Lothiana, for the moment, considered a reptiliomorph rather than a true amniot, and Paleogyrus, both of similar build and presumably similar habit. Rise of the reptiles. The earliest amniots, including stem reptiles, those amniots closer to modern reptiles than to mammals, were largely overshadowed by larger stem tetrapods, such as Cochleosaurus, and remained a small, inconspicuous part of the fauna until the Carboniferous rainforest collapse. This sudden collapse affected several large groups. Primitive tetrapods were particularly devastated, while stem reptiles fared better, being ecologically adapted to the drier conditions that followed. Primitive tetrapods, like modern amphibians, need to return to water to lay eggs. In contrast, amniots, like modern reptiles, whose eggs possess a shell that allows them to be laid on land, were better adapted to the new conditions. Amniots acquired new niches at a faster rate than before the collapse and at a much faster rate than primitive tetrapods. They acquired new feeding strategies including herbivory and carnivory, previously only having been insectivores and piscivores. From this point forward, reptiles dominated communities and had a greater diversity than primitive tetrapods, setting the stage for the Mesozoic, known as the Age of Reptiles. One of the best-known early stem reptiles is Mesosaurus, a genus from the early Permian that had returned to water, feeding on fish, anapsids, synapsids, diapsids, and sauropsids. It was traditionally assumed that the first reptiles retained an inapsid skull inherited from their ancestors. This type of skull has a skull roof with only holes for the nostrils, eyes and pineal. The discoveries of synapsid-like openings, see below, in the skull roof of the skulls of several members of Parareptilia. The clade containing most of the amniotes traditionally referred to as inapsids, including lanthanosuchoids, milleretids, bolosaurids, some nycterolitirids, some procolophenoids and at least some mesosaurs made it more ambiguous, and it's currently uncertain whether the ancestral amniot had an anapsid like a synapsid-like skull. These animals are traditionally referred to as inapsids, and form a paraphyletic basic stock from which other groups evolved. Very shortly after the first amniots appeared, a lineage called synapsida split off. This group was characterized by a temporal opening in the skull behind each eye to give room for the jaw muscle to move. These are the mammal-like amniots, or stem mammals, that later gave rise to the true mammals. Soon after, another group evolved a similar trait, this time with a double opening behind each eye, earning them the name Dipsida, two arches. The function of the holes in these groups was to lighten the skull and give room for the jaw muscles to move, allowing for a more powerful bite. Turtles have been traditionally believed to be surviving parareptiles, on the basis of their anapsid skull structure, which was assumed to be primitive trait. The rationale for this classification has been disputed, with some arguing that turtles are diapsids that evolved anapsid skulls in order to improve their armor. Later morphological phylogenetic studies with this in mind place turtles firmly within Dipsida. All molecular studies have strongly upheld the placement of turtles within diapsids, most commonly as a sister group to extant archosaurs, Permian reptiles. With the close of the Carboniferous, the amniots became the dominant tetrapod fauna, while primitive, terrestrial reptiliomorphs still existed. The synapsid amniots evolved the first truly terrestrial megafauna, giant animals, in the form of pelycosaurs, such as Edaphosaurus and the carnivorous Dimetrodon. In the mid-Permian period, the climate became drier, resulting in a change of fauna. The pelycosaurs were replaced by the theropods, the parareptiles, whose massive skull roofs had no post-orbital holes, continued and flourished throughout the Permian. The Parasaurian parareptiles reached giant proportions in the late Permian, eventually disappearing at the close of the period, the turtles being possible survivors. Early in the period, the modern reptiles, or crown group reptiles, evolved and split into two main lineages. The Archosauromorpha, forebears of turtles, crocodiles, and dinosaurs, and the Lepidosauromorpha, predecessors of modern lizards and tuataras. Both groups remained lizard-like and relatively small and inconspicuous during the Permian. Mesozoic reptiles. The close of the Permian saw the greatest mass extinction known. See the Permian Triassic extinction event. An event prolonged by the combination two or more distinct extinction pulses. Most of the earlier parareptile and synapsid megafauna disappeared, being replaced by the true reptiles, particularly Archosauromorphs. These were characterized by elongated hind legs and an erect pose, the early forms looking somewhat like long-legged crocodiles. The archosaurs became the dominant group during the Triassic period, though it took 30 million years before their diversity was as great as the animals that lived in the Permian. Archosaurs developed into the well-known dinosaurs and pterosaurs, as well as the ancestors of crocodiles. Since reptiles, first Velociraptors and then dinosaurs, dominated the Mesozoic era, the interval is popularly known as the Age of Reptiles. The dinosaurs also developed smaller forms, including the feather-bearing smaller theropods. In the Cretaceous period, these gave rise to the first true birds. The sister group Tuatara is Lepidosauромorpha, containing lizards and tuataras, as well as their fossil relatives. Lepidosauромorpha contained at least one major group of the Mesozoic sea reptiles, the mosasaurs, which lived during the Cretaceous period. The phylogenetic placement of other main groups of fossil sea reptiles, the ichthyopterygians, including ichthyosaurs, and the sauropterygians, which evolved in the early Triassic, is more controversial. Different authors linked these groups either to Lepidosauромorphs or to Archosauromorphs, and ichthyopterygians were also argued to be diapsids that did not belong to the least inclusive clade containing Lepidosauромorphs and Archosauromorphs. Cenozoic reptiles. The close of the Cretaceous period saw the demise of the Mesozoic-era reptilian megafauna. See the Cretaceous-Paleogene extinction event. Of the large marine reptiles, only sea turtles were left, and of the non-marine large reptiles, only the semi-aquatic crocodiles and broadly similar choristidés survived the extinction, with the latter becoming extinct in the Miocene. Of the great host of dinosaurs dominating the Mesozoic, only the small-beaked birds survived. This dramatic extinction pattern at the end of the Mesozoic led into the Cenozoic. Mammals and birds filled the empty niches left behind by the reptilian megafauna and, while reptile diversification slowed, bird and mammal diversification took an exponential turn. However, reptiles were still important components of the megafauna, particularly in the form of giant tortoises. After the extinction of most archosaur and marine reptile lines by the end of the Cretaceous, reptile diversification continued throughout the Cenozoic. Squamates took a massive hit during the KT event, only recovering 10 million years after it. But they underwent a great radiation event once they recovered, and today squamates make up the majority of living reptiles, greater than 95%. Approximately 10,000 extant species of traditional reptiles are known, with birds adding about 10,000 more, almost twice the number of mammals, represented by about 5,700 living species, excluding domesticated species. Morphology and physiology. Circulation. All squamates and turtles have a three-chambered heart consisting of two atria, one variably partitioned ventricle, and two aortas that lead to the systemic circulation. The degree of mixing of oxygenated and deoxygenated blood in the three-chambered heart varies, depending on the species and physiological state. Under different conditions, deoxygenated blood can be shunted back to the body or oxygenated blood can be shunted back to the lungs. This variation in blood flow has been hypothesized to allow more effective thermoregulation and longer diving times for aquatic species, but has not been shown to be a fitness advantage. For example, iguana hearts, like the majority of the squamates' hearts, are composed of three chambers with two aorta and one ventricle. Cardiac and voluntary muscles. The main structures of the heart are the sinus venosus, the pacemaker, the left atrium, the right atrium, the atrioventricular valve, the cavum venosum, cavum arteriosum, the cavum pulmonale, the muscular ridge, the ventricular ridge, pulmonary veins, and paired aortic arches. Some squamate species, e.g., pythons and monitor lizards, have three-chambered hearts that become functionally four-chambered hearts during contraction. This is made possible by a muscular ridge that subdivides the ventricle during ventricular diastole and completely divides it during ventricular systole. Because of this ridge, some of these squamates are capable of producing ventricular pressure differentials that are equivalent to those seen in mammalian and avian hearts. Crocodilians have an anatomically four-chambered heart. similar to birds, but also have two systemic aortas and are therefore capable of bypassing their pulmonary circulation. Metabolism. Modern non-avian reptiles exhibit some form of cold-bloodedness, i.e., some mix of poikilothermy, ectothermy, and bradymetabolism, so that they have limited physiological means of keeping the body temperature constant and often rely on external sources of heat, due to a less stable core temperature than birds and mammals. Reptilian biochemistry requires enzymes capable of maintaining efficiency over a greater range of temperatures than in the case for warm-blooded animals. The optimum body temperature range varies with species, but is typically below that of warm-blooded animals. For many lizards, it falls in the 24 degrees to 35 degrees Celsius, 75 degrees to 95 degrees Fahrenheit range, while extreme heat-adapted species, like the American desert iguana Dipsosaurus dorsalis, can have optimal physiological temperatures in the mammalian range, between 35 degrees and 40 degrees Celsius, 95 degrees and 104 degrees Fahrenheit, while the optimum temperature is often encountered when the animal is active. The low basal metabolism makes body temperature drop rapidly when the animal is inactive. As in all animals, reptilian muscle action produces heat. In large reptiles, like leatherback turtles, the low surface-to-volume ratio allows this metabolically produced heat to keep the animals warmer than their environment, even though they do not have a warm-blooded metabolism. This form of homeothermy is called gigantothermy. It has been suggested as having been common in large dinosaurs and other extinct large-bodied reptiles. The benefit of a low resting metabolism is that it requires far less fuel to sustain bodily functions. By using temperature variations in their surroundings, or by remaining cold when they do not need to move, reptiles can save considerable amounts of energy compared to endothermic animals of the same size. A crocodile needs from a tenth to a fifth of the food necessary for a lion of the same weight and can live half a year without eating. Lower food requirements and adaptive metabolisms allow reptiles to dominate the animal life in regions where net calorie availability is too low to sustain large-bodied mammals and birds. It is generally assumed that reptiles are unable to produce the sustained high-energy output necessary for long-distance chases or flying. Higher energetic capacity might have been responsible for the evolution of warm-bloodedness in birds and mammals. However, investigation of correlations between active capacity and thermophysiology show a weak relationship. Most extant reptiles are carnivores with a sit-and-wait feeding strategy. Whether reptiles are cold-blooded due to their ecology is not clear. Energetic studies on some reptiles have shown active capacities equal to or greater than similar sized warm-blooded animals. Respiratory system. All reptiles breathe using lungs. Aquatic turtles have developed more permeable skin, and some species have modified their cloaca to increase the area for gas exchange. Even with these adaptations, breathing is never fully accomplished without lungs. Lung ventilation is accomplished differently in each main reptile group. In squamates, the lungs are ventilated almost exclusively by the axial musculature. This is also the same musculature that is used during locomotion. Because of this constraint, most squamates are forced to hold their breath during intense runs. Some, however, have found a way around it. The ranids, and a few other lizard species, employ buccal pumping as a complement to their normal axial breathing. This allows the animals to completely fill the lungs during intense locomotion, and thus remain aerobically active for a long time. Tegu lizards are known to possess a proto-diaphragm. which separates the pulmonary cavity from the visceral cavity. While not actually capable of movement, it does allow for greater lung inflation by taking the weight of the viscera off the lungs. Crocodilians actually have a muscular diaphragm that is analogous to the mammalian diaphragm. The difference is that the muscles for the crocodilian diaphragm pull the pubis, part of the pelvis, which is movable in crocodilians, back, which brings the liver down, thus freeing space for the lungs to expand. This type of diaphragmatic setup has been referred to as the hepatic piston. The airway's bronchia form a number of double tubular chambers within each lung. On inhalation and exhalation, air moves through the airways in the same direction, thus creating a unidirectional airflow through the lungs. A similar system is found in birds, monitor lizards and iguanas. Most reptiles lack a secondary palate, meaning that they must hold their breath while swallowing. Crocodilians have evolved a bony secondary palate that allows them to continue breathing while remaining submerged, and protect their brains against damage by struggling prey. Skinks, family Cincidae, also have evolved a bony secondary palate. To varying degrees, snakes took a different approach and extended their trachea instead. Their tracheal extension sticks out like a fleshy straw, and allows these animals to swallow large prey without suffering from asphyxiation. Turtles and tortoises equals. How turtles and tortoises breathe has been the subject of much study. To date, only a few species have been studied thoroughly enough to get an idea of how those turtles breathe. The varied results indicate that turtles and tortoises have found a variety of solutions to this problem. The difficulty is that most turtle shells are rigid and do not allow for the type of expansion and contraction that other amniotes use to ventilate the lungs. Some turtles, such as the Indian flap shell, Lysimys punctata, have a sheet of muscle that envelops the lungs. When it contracts, the turtle can exhale. When at rest, the turtle can retract the limbs into the body cavity and force air out of the lungs. When the turtle protracts its limbs, the pressure inside the lungs is reduced, and the turtle can suck air in. Turtle lungs are attached to the inside of the top of the shell carapace, with the bottom of the lungs attached, via connective tissue, to the rest of the viscera, by using a series of special muscles, roughly equivalent to a diaphragm. Turtles are capable of pushing their viscera up and down, resulting in effective respiration, since many of these muscles have attachment points in conjunction with their forelimbs. Indeed, many of the muscles expand into the limb pockets during contraction. Breathing during locomotion has been studied in 3 species, and they show different patterns. Adult female green sea turtles do not breathe as they crouch along their nesting beaches. They hold their breath during terrestrial locomotion and breathe in bouts as they rest. North American box turtles breathe continuously during locomotion, and the ventilation cycle is not coordinated with the limb movements. This is because they use their abdominal muscles to breathe during locomotion. The last species to have been studied is the red-eared slider, which also breathes during locomotion, but takes smaller breaths during locomotion than during small pauses between locomotor bouts, indicating that there may be mechanical interference between the limb movements and the breathing apparatus. Box turtles have also been observed to breathe while completely sealed up inside their shells. Skin. Reptilian skin is covered in a horny epidermis, making it watertight and enabling reptiles to live on dry land. In contrast to amphibians, compared to mammalian skin, that of reptiles is rather thin and lacks the thick dermal layer that produces leather in mammals. Exposed parts of reptiles are protected by scales or scutes, sometimes with a bony base, forming armor. In lepidosaurians, such as lizards and snakes. The whole skin is covered in overlapping epidermal scales. Such scales were once thought to be typical of the class Reptilia as a whole, but are now known to occur only in lepidosaurians. The scales found in turtles and crocodiles are of dermal, rather than epidermal, origin and are properly termed scutes. In turtles, the body is hidden inside a hard shell composed of fused scutes, lacking a thick dermis. Reptilian leather is not as strong as mammalian leather. It is used in leatherwares for decorative purposes for shoes, belts and handbags, particularly crocodile skin. Shedding. Reptiles shed their skin through a process called ecdysis, which occurs continuously throughout their lifetime. In particular, younger reptiles tend to shed once every 5 to 6 weeks, while adults shed 3 to 4 times a year. Younger reptiles shed more because of their rapid growth rate. Once full size, the frequency of shedding drastically decreases. The process of ecdysis involves forming a new layer of skin under the old one. Proteolytic enzymes and lymphatic fluid is secreted between the old and new layers of skin. Consequently, this lifts the old skin from the new one, allowing for shedding to occur. Snakes will shed from the head to the tail, while lizards shed in a patchy pattern. Dysectasis, a common skin disease in snakes and lizards, will occur when ecdysis, or shedding, fails. There are numerous reasons why shedding fails and can be related to inadequate humidity and temperature, nutritional deficiencies, dehydration and traumatic injuries. Nutritional deficiencies decreases proteolytic enzymes, while dehydration reduces lymphatic fluids to separate the skin layers. Traumatic injuries, on the other hand, form scars that will not allow new scales to form and disrupt the process of ecdysis. Excretion. Excretion is performed mainly by 2 small kidneys. In diapsids, uric acid is the main nitrogenous waste product. Turtles, like mammals, excrete mainly urea. Unlike the kidneys of mammals and birds, reptile kidneys are unable to produce liquid urine more concentrated than their body fluid. This is because they lack a specialized structure called a loop of Henle, which is present in the nephrons of birds and mammals. Because of this, many reptiles use the colon to aid in the reabsorption of water. Some are also able to take up water stored in the bladder. Excess salts are also excreted by nasal and lingual salt glands in some reptiles. Digestion. Most reptiles are insectivorous or carnivorous and have rather simple and comparatively short digestive tracts, meat being fairly simple to break down and digest. Digestion is slower than in mammals, reflecting their lower resting metabolism and their inability to divide and masticate their food. Their poikilotherm metabolism has very low energy requirements, allowing large reptiles like crocodiles and the large constrictors to live from a single large meal for months, digesting it slowly. While modern reptiles are predominantly carnivorous, during the early history of reptiles several groups produced some herbivorous megafauna. In the Paleozoic, the parasaurs, and in the Mesozoic several lines of dinosaurs. Today, the turtles are the only predominantly herbivorous reptile group, but several lines of agamas and iguanas have evolved to live wholly or partly on plants. Herbivorous reptiles face the same problems of mastication as herbivorous mammals but lacking the complex teeth of mammals. Many species swallow rocks and pebbles, so-called gastroliths, to aid in digestion. The rocks are washed around in the stomach, helping to grind up plant matter. Fossil gastroliths have been found associated with both ornithopods and sauropods, though whether they actually functioned as a gastric mill in the latter is disputed. Saltwater crocodiles also use gastroliths as ballast, stabilizing them in the water or helping them to dive. A dual function as both stabilizing ballast and digestion aid has been suggested for gastroliths found in plesiosaurs. Nerves. The reptilian nervous system contains the same basic part of the amphibian brain, but the reptile cerebrum and cerebellum are slightly larger. Most typical sense organs are well developed with certain exceptions, most notably the snake's lack of external ears. Middle and inner ears are present. There are 12 pairs of cranial nerves. Due to their short cochlea, reptiles use electrical tuning to expand their range of audible frequencies. Intelligence equals. Reptiles are generally considered less intelligent than mammals and birds. The size of their brain relative to their body is much less than that of mammals, the encephalization quotient being about 1/10 of that of mammals, though larger reptiles can show more complex brain development. Larger lizards, like the monitors, are known to exhibit complex behavior, including cooperation. Crocodiles have relatively larger brains and show a fairly complex social structure. The Komodo dragon is even known to engage in play, as are turtles, which are also considered to be social creatures and sometimes switch between monogamy and promiscuity in their sexual behavior. One study found that wood turtles were better than white rats at learning to navigate mazes. Vision. Most reptiles are diurnal animals. The vision is typically adapted to daylight conditions, with color vision and more advanced visual depth perception than in amphibians and most mammals. In some species, such as blind snakes, vision is reduced. Some snakes have extra sets of visual organs, in the loosest sense of the word, in the form of pits sensitive to infrared radiation. Heat. Such heat-sensitive pits are particularly well developed in the pit vipers, but are also found in boas and pythons. These pits allow the snakes to sense the body heat of birds and mammals, enabling pit vipers to hunt rodents in the dark. Reproduction. Reptiles generally reproduce sexually, though some are capable of asexual reproduction. All reproductive activity occurs through the cloaca, the single exit entrance at the base of the tail where waste is also eliminated. Most reptiles have copulatory organs, which are usually retracted or inverted and stored inside the body. In turtles and crocodilians, the male has a single median penis, while squamates, including snakes and lizards, possess a pair of hemipenes, only one of which is typically used in each session. Tuatara, however, lack copulatory organs, and so the male and female simply press their cloacas together as the male discharges sperm. Most reptiles lay amniotic eggs covered with leathery or calcareous shells. An amnion, chorion, and allantois are present during embryonic life. The eggshell, 1, protects the crocodile embryo, 11, and keeps it from drying out, but it is flexible to allow gas exchange. The chorion, 6, aids in gas exchange between the inside and outside of the egg. It allows carbon dioxide to exit the egg and oxygen gas to enter the egg. The albumen, 9, further protects the embryo and serves as a reservoir for water and protein. The allantois, 8, is a sac that collects the metabolic waste produced by the embryo. The amniotic sac, 10, contains amniotic fluid, 12, which protects and cushions the embryo. The amnion, 5, aids in osmoregulation and serves as a saltwater reservoir. The yolk sac, 2, surrounding the yolk, 3, contains protein and fat-rich nutrients that are absorbed by the embryo via vessels, 4, that allow the embryo to grow and metabolize. The air space, 7, provides the embryo with oxygen while it is hatching. This ensures that the embryo will not suffocate while it is hatching. There are no larval stages of development. Viviparity and ovoviviparity have evolved in many extinct clades of reptiles and in squamates. In the latter group, many species, including all boas and most vipers, utilize this mode of reproduction. The degree of viviparity varies. Some species simply retain the eggs until just before hatching. Others provide maternal nourishment to supplement the yolk. And yet others lack any yolk and provide all nutrients via a structure similar to the mammalian placenta. The earliest documented case of viviparity in reptiles is the early Permian mesosaurs. Although some individuals are taxa in that clade may also have been oviparous because a putative, isolated egg has also been found. Several groups of Mesozoic marine reptiles also exhibited viviparity, such as mosasaurs, ichthyosaurs, and sauropterygia, a group that include pachypleurosaurs and plesiosauria. Asexual reproduction has been identified in squamates in 6 families of lizards and 1 snake. In some species of squamates, a population of females is able to produce a unisexual diploid clone of the mother. This form of asexual reproduction, called parthenogenesis, occurs in several species of gecko, and is particularly widespread in the teiids, especially Aspidocellus, and lacertids. Lacerta. In captivity, Komodo dragons, Varanidae, have reproduced by parthenogenesis. Parthenogenetic species are suspected to occur among chameleons, agamids, xanthusiids, and typhlopids. Some reptiles exhibit temperature-dependent sex determination, TDSD, in which the incubation temperature determines whether a particular egg hatches as male or female. TDSD is most common in turtles and crocodiles, but also occurs in lizards and tuatara. To date, there has been no confirmation of whether TDSD occurs in snakes. Defense mechanisms. Many small reptiles, such as snakes and lizards that live on the ground or in the water, are vulnerable to being preyed on by all kinds of carnivorous animals. Thus avoidance is the most common form of defense in reptiles. At the first sign of danger, most snakes and lizards crawl away into the undergrowth, and turtles and crocodiles will plunge into water and sink out of sight. Camouflage and warning. Reptiles tend to avoid confrontation through camouflage. 2 major groups of reptile predators are birds and other reptiles, both of which have well-developed color vision. Thus the skins of many reptiles have cryptic coloration of plain or mottled gray, green, and brown to allow them to blend into the background of their natural environment. Aided by the reptile's capacity for remaining motionless for long periods, the camouflage of many snakes is so effective that people or domestic animals are most typically bitten because they accidentally step on them. When camouflage fails to protect them, blue-tongued skinks will try to ward off attackers by displaying their blue tongues, and the frill-necked lizard will display its brightly colored frill. These same displays are used in territorial disputes and during courtship, if danger arises so suddenly that flight is useless. Crocodiles, turtles, some lizards, and some snakes hiss loudly when confronted by an enemy. Rattlesnakes rapidly vibrate the tip of the tail, which is composed of a series of nested, hollow beads to ward off approaching danger. In contrast to the normal drab coloration of most reptiles, the lizards of the genus Heloderma, the Gila monster and the beaded lizard, and many of the coral snakes have high contrast warning coloration, warning potential predators they are venomous. A number of non-venomous North American snake species have colorful markings similar to those of the coral snake, an oft-cited example of Batesian mimicry. Alternative defense in snakes. Camouflage will not always fool a predator. When caught out, Snake species will adopt different defensive tactics and use a complicated set of behaviors when attacked. Some will first elevate their head and spread out the skin of their neck in an effort to look large and threatening. Failure of this strategy may lead to other measures practiced particularly by cobras, vipers, and closely related species, who use venom to attack. The venom is modified saliva, delivered through fangs from a venom gland. Some non-venomous snakes, such as American hognose snakes or European grass snake, play dead when in danger. Defense in crocodilians. When a crocodilian is concerned about its safety, it will gape to expose the teeth and yellow tongue. If this doesn't work, the crocodilian gets a little more agitated and typically begins to make hissing sounds. After this, the crocodilian will start to change its posture dramatically to make itself look more intimidating. The body is inflated to increase apparent size. If absolutely necessary, it may decide to attack an enemy. Some species try to bite immediately. Some will use their heads as sledgehammers and literally smash an opponent. Some will rush or swim toward the threat from a distance, even chasing the opponent onto land or galloping after it. The main weapon in all crocodiles is the bite, which can generate very high bite force. Many species also possess canine-like teeth. These are used primarily for seizing prey, but are also used in fighting and display, shedding and regenerating tails. Geckos, skinks, and other lizards that are captured by the tail will shed part of the tail structure through a process called autotomy and thus be able to flee. The detached tail will continue to wiggle, creating a deceptive sense of continued struggle and distracting the predator's attention from the fleeing prey animal. The detached tails of leopard geckos can wiggle for up to 20 minutes. In many species, the tails are of a separate and dramatically more intense color than the rest of the body so as to encourage potential predators to strike for the tail first. In the shingleback skink and some species of geckos, the tail is short and broad and resembles the head, so that the predators may attack it rather than the more vulnerable front part. Reptiles that are capable of shedding their tails can partially regenerate them over a period of weeks. The new section will however contain cartilage rather than bone, and will never grow to the same length as the original tail. It is often also distinctly discolored compared to the rest of the body and may lack some of the external sculpting features seen in the original tail. Relations with humans. In cultures and religions, dinosaurs have been widely depicted in culture since the English paleontologist Richard Owen coined the name dinosaur in 1842. As soon as 1854, the Crystal Palace dinosaurs were on display to the public in South London. One dinosaur appeared in literature even earlier, as Charles Dickens placed a megalosaurus in the first chapter of his novel Bleak House in 1852. The dinosaurs featured in books, films, television programs, artwork, and other media have been used for both education and entertainment. The depictions range from the realistic, as in the television documentaries of the 1990s and first decade of the 21st century, or the fantastic, as in the monster movies of the 1950s and 1960s. The snake or serpent has played a powerful symbolic role in different cultures. In Egyptian history, the Nile cobra adorned the crown of the pharaoh. It was worshipped as one of the gods and was also used for sinister purposes. Murder of an adversary and ritual suicide. Cleopatra. In Greek mythology, snakes are associated with deadly antagonists. As a chthonic symbol, roughly translated as earthbound, the nine-headed Lernaean hydra that Hercules defeated and the three Gorgon sisters are children of Gaia, the earth. Medusa was one of the three Gorgon sisters who Perseus defeated. Medusa is described as a hideous mortal, with snakes instead of hair and the power to turn men to stone with her gaze. After killing her, Perseus gave her head to Athena, who fixed it to her shield called the Aegis. The Titans are depicted in art with their legs replaced by bodies of snakes for the same reason. They are children of Gaia, so they are bound to the earth. In Hinduism, snakes are worshipped as gods, with many women pouring milk on snake pits. The cobra is seen on the neck of Shiva, while Vishnu is depicted often as sleeping on a seven-headed snake or within the coils of a serpent. There are temples in India solely for cobras, sometimes called Nagraj, king of snakes, and it is believed that snakes are symbols of fertility. In the annual Hindu festival of Nag Panchami, snakes are venerated and prayed to. In religious terms, the snake and jaguar are arguably the most important animals in ancient Mesoamerica. In states of ecstasy, lords dance a serpent dance. Great descending snakes adorn and support buildings from Chichen Itza to Tenochtitlan. And the Nahuatl word coatl, meaning serpent or twin, forms part of primary deities such as Mixcoatl, Calcotl, and Coatlicue. Quote, in Christianity and Judaism, a serpent appears in Genesis to tempt Adam and Eve with a forbidden fruit from the tree of knowledge of good and evil. The turtle has a prominent position as a symbol of steadfastness and tranquility in religion, mythology, and folklore from around the world. A tortoise's longevity is suggested by its long lifespan and its shell, which was thought to protect it from any foe. In the cosmological myths of several cultures, a world turtle carries the world upon its back or supports the heavens. Medicine. Deaths from snake bites are uncommon in many parts of the world, but are still counted in tens of thousands per year in India. Snakebite can be treated with antivenom made from the venom of the snake. To produce antivenom, a mixture of the venoms of different species of snake is injected into the body of a horse in ever-increasing dosages until the horse is immunized. Blood is then extracted. The serum is separated, purified and freeze-dried. The cytotoxic effect of snake venom is being researched as a potential treatment for cancers. Geckos have also been used as medicine, especially in China. Other crocodiles are protected in many parts of the world and farmed commercially. Their hides are tanned and used to make leather goods such as shoes and handbags. Crocodile meat is also considered a delicacy. The most commonly farmed species are the saltwater and Nile crocodiles. Farming has resulted in an increase in the saltwater crocodile population in Australia, as eggs are usually harvested from the wild, so landowners have an incentive to conserve their habitat. Crocodile leather is made into wallets, briefcases, purses, handbags, belts, hats, and shoes. Crocodile oil has been used for various purposes. In the Western world, some snakes, especially docile species such as the ball python and corn snake, are kept as pets. See also, evolution of reptiles, herpetophobia, list of reptiles, lists of reptiles by region, list of threatened reptiles and amphibians of the United States. Ophidiophobia. Reptile database. Further reading. Notes. Data related to Reptilia at Wikispecies. Reptilia at Wikibooks. Reptile phylogeny. Reptile images. Sri Lanka Wildlife Information Database. Biology of the Reptilia is an online copy of the full text of a 22-volume 13,000-page summary of the state of research of reptiles.